So, good morning, everyone. Now it's my turn to present our group, Animal-Microbiota Interactions. My talk is going to be considerably less, lower resolution, and probably shorter than chance. But I will be introducing what we do in our group. That we are all these people, and a couple of more people that are also working that are missing in that group meeting. We all can say that was June probably because we were wearing winter clothes. But Carlotta was still pregnant. Now she has a baby, and Isolde was also pregnant, and now she has a baby. So we are increasing the size of our group considerably. So it's 2 kids per month or something like that. So I will be mainly talking about the 2 main projects our group's activity is based on. So one is the Earthology initiative, and the other one is metagenomics. And they both tackle animal-microbiota interactions, but from very different perspectives and using quite different methodologies. And you will see that there are many other smaller projects, but they are all somehow related to these 2 projects. So regarding the Earthology initiative, I will be giving you an overview, but there is a website with a lot of information that if you're interested, you can find more info there. And all these people are intensively working this project. Ella managing it. Aoife used to be the main manager. Now Ella is working there. Nana is the main person processing thousands of samples. And Carlotta is off, but Rafael is being responsible for setting up all the bioinformatics regarding the EHI. And then Jonas has been focused mainly on translating all these procedures that we've been implementing in the labs to the automation realm. And so this is the map of the collaborators of the Earthology initiative. We are not alone at all. We are over 200 researchers all over the world. You can see that we have also oversampled Europe. But the reason was that we have started in Europe, and because we had to test all the management administrative hurdles of transferring thousands of biological samples from all over the world to here in Denmark, because we are doing everything following very strict standardized procedures. So what we do is we contact, or we are contacted by people all over the world, and then we agree what kind of sampling is going to happen, and based on that information we prepare these sampling kits, and those sampling kits contain tubes that are already pre-filled with a standardized buffer. They're also labeled using the QR codes that we are using them here in the labs to keep everything organized, because that's one of the main points. It is thousands and thousands of samples, and they can be from very different sources, and so we need to keep all this organized, otherwise it's a big nightmare. And then all the samples need to come with standardized metadata as well, because then we want to make large analysis that will incorporate all this information into the models that we will be building, and that requires that all the ontology of the metadata is properly standardized, so then we can actually match the data with the environmental information, with the animal-related information, etc. I didn't mention that the Earthology initiative stands for the hologenomics. In this case, we are working on vertebrates and their gut microbiomes, and that's why it is very important for us to get metadata from the individual animals as well as the environment, because we have kind of a 2-layer the environment there. We need to know whether the individual animal, what was the weight, the size, whether it had any signs of sickness or whatever, and then we need to know also what environment this animal was trapped, to then collect the fecal samples that we use as the primary source of both microbial DNA as well as host DNA. So we use feces as the primary source, but we also request the collaborators to provide us with alternative samples. If there is not enough host DNA in the feces, we can extract DNA from saliva, from blood, from tissue, and that way we can generate not only microbial metagenomic data, but also host genomic data for doing different analysis of population genetics, or also analysis of adaptation signatures to environmental gradients, and so on and so forth. So what we basically do in this EHI is to generate a lot of data, and then we apply genomes of metagenomics. What we try to do is to build the best metagenomic assemblies that we can, and then reconstruct bacterial genome from those assemblies. And so for the many projects we are running, we generate this kind of collections of bacterial genomes that we annotate, and then we have developed tools to extract information from the raw gene annotations and to create this kind of more meaningful data sets that inform us about the functional capabilities of the bacteria. So this way, in this here, you can see the phylogeny of this bacteria from probably some kind of amphibian, perhaps, but could be. And then here, what you can see is each of these columns is a functional trait that we have distilled from the gene annotations of the bacteria. So this way, we can see, for instance, that the functional attributes of this guy here, you can see that it is quite wide. It's quite different to the one that is close to it, which is phylogenetically quite similar, but functionally being different. And we, of course, need to account for the completeness and the contamination of the genomes, but we also try to incorporate our models so that the quality of the genome reconstruction affects the least possible to the functional inferences that we do. So we do this for many projects, right? And many species. So far, we have received samples from over 200 species, but 144 are more or less okay-ish characterized. We have processed over almost 2,600 samples. So Nana has. So these are the ones that I think reached the final destination. So you can imagine that quite a few were left away because they were not high enough quality. So you cannot imagine how, even though everything is standardized, how non-standardized the samples that we get could be. So we also need to do some filtering. So far, we have generated over now 15 terabases of data, and that has yielded now over 50,000 bacterial genomes associated with this 144 vertebrate species. And this goes over 15 million genes that are encapsulated in all these bacterial genomes and that have been annotated and so on and so forth. What we do in the EHI is to apply these standardized methodologies for collecting the samples, for generating the DNA structure, library preparation, all the bioinformatics. Everything is standardized, but using this baseline, we run different projects with different hypotheses, different study aims, different experimental designs, etc., with the aim that we will be able to address specific questions for the given study system, but then we will be able to reuse this data to address other questions that encompass multiple projects. And here we have some examples. For instance, Garasi, a PhD student, is working on elevation hologenomics, where she is working with lizard samples in 4 transects in the Pyrenees, going from down 400 meters of elevation to over 2,000 meters of elevation, and seeing how this altitudinal gradients, or how the different populations change in terms of genomic and metagenomic signatures of elevational change in the Pyrenees. Claudia, Marie Curie postdoc, is working on invasion hologenomics, which is a project in which she is comparing the, so far it's been just the microbial aspect of it, but she's comparing how the adaptation to urban or anthropomorphized environments changes the microbiome in gray squirrels, which are invasive in Italy, and the native red squirrels. And the results are quite cool. I won't show you the results because I didn't have time, but the results are really cool because they really behave in different ways. And as you can imagine, the invasive one is working. So, and then Isolde, who is in maternity leave, but she will be back after summer, is working on another Marie Curie project. In this case, is on chytrid fungus, bombina toads, and their genomics, and the microbial communities, but in this case, it's in skin microbiome. And it is really cool because in the beginning, we really struggled to get high-quality DNA and libraries and bacterial genomes from the bombina toads because the traditional ways to sample the skin, etc., were not really working that well. But we did many tests, and now we have a catalog of almost 300 bacterial genomes from the skin of the bombinas, which is really cool. They are very different, and bombinas are cool. I don't know if you haven't seen them. They are black on yellow and red. And of course, we always have logos for the. That's very important. That's the first point. Let's create a logo, and then we think about the rest. And then, as I mentioned, what we aim to do once we have all these isolated projects is to reuse all this data to address other questions, and that are encompassing multiple bacteria, multiple hosts, etc., etc. And right now, David is working on his master's project, mainly focused on genome-reduced bacteria. So these are weird bacteria that have extremely reduced genomes that appear in all vertebrates, basically, that we don't really know why, how they are living this way and how they are that widespread. So David is reusing all this information, and he will need to hand in. Where is David? David is there. Yeah. So he will need to hand it by September. So it's gonna be cool to see how it's gonna be the first attempt to put all this information together, and then bigger projects will come later. And Johan, right now a student assistant, is collecting a lot of metadata of life history traits of the hosts of the animals, and creating a huge database with information on diets, metabolism, sociality, and other aspects that we know that can affect the microbiome. And the idea is then that we don't only analyze the microbiome changes across hosts based on the phylogenies of the host, but also the ecological traits. So to see whether and how different microbes have diversified across different hosts, depending on what are the ecological evolutionary attributes of the host themselves. And of course, not everything, but a huge part of it is based on Airtable. It's the second, let's say, wife of Bent. So he has his wife, and then it's Airtable, and then right, Airtable, and then that's right. And so we use Airtable pretty much for everything. And so if you have any interest, if you have ever heard, I am more than willing to, and I'm sure that Bent is as well.
Is it not the same for you as well? It's also your second wife.
She's here, so I won't.
No, that's why.
But definitely, I spend more time. But what we do with this is not just to collect all the metadata, but we have set up mainly Rafael has done an awesome job in connecting Airtable with Mjolnir, and Mjolnir with Erda, and Erda with Airtable. So we have the trio here that's connect with each other. And pretty much we don't need to do anything. It's kind of 10 PhD students in one that is doing everything, but very large. So it's been a huge effort of setting up all this. We've been working for almost 4 years without really having any publication at all. So I really appreciate the work that everyone has done, because it's not easy to keep the faith and things that will work, because it takes a lot of time, a lot of effort to set all this up. But finally, as Morten mentioned before, the last week or 2 weeks ago, the reference paper for the EHA was published in Trends in Ecology and Evolution. And then we have 3 more papers that are dealing with the technical aspects of it that we really hope we will submit before June. So that's the deadline. So these 2 papers are almost ready. 10 days. And then we will also have our first data release after summer. So we have a strategy to releasing all this data. We need to do it in a coherent way so that it is feasible to put all the data in the public archives, but also with all the metadata, etc. But then people can really fetch that information properly. So yeah, these are the first outcomes that will come. And then we are working on many manuscripts in all these projects, and hopefully by the end of the year we can submit a handful of those. So that'd be good. And the other one is 3D-omics, and this is a European project similar to Pino. And in here, we have many other people working. Again, you can see some of the faces that work in the EHI, because some people have been jumping between the projects. And in here, we collaborate mainly with people from the section for microbiology in the Faculty of Science. And it's Ulrich's team with Brian and Zoe, who are working mainly on the imaging aspect of it. They're doing fluorescence in situ hybridization methods and improving them, etc. And then we have here, so Carlotta has been working on this for over 2 years. Then Amalia, joined us in February. And Jorge, so Amalia has been mainly working so far in the lab work with Jonas and Nana. And then Jorge has been in charge of setting up all the bioinformatics suite of tools and that we've been using for generating, again, tons of data. The idea behind 3D Omics is that when we do conventional metagenomics, we get some bulk samples, even though they might be very small. It can be a fecal pellet, it can be intestinal content, but what we do is to homogenize the sample. And we know that when we homogenize the sample, we are losing a lot of information. Because in ecology, in any kind of interactions between organisms, we know space matters. Time matters, but the space also matters. And so far, we've been completely neglecting the spatial aspect. So we might have two bacteria that were, on our scale, thousands of kilometers away in an intestinal environment, and we were considering them as to be together. So now we are trying to resolve that by doing microbial spatial metagenomics at the scale in which microorganisms interact. And we are doing that, so you can see here an example. This is intestinal crosscut of the cecum of a chicken. So this is 1 centimeter, more or less. So this is this size. And then, I don't know if you can see these small holes here. So these are the actual samples that we are using for generating the microbial communities. And this is, if we do fish on this, if we stain the bacteria, this is how they look like, more or less. We have 4 here that have been stained with different colors. And the size of the sample is always between 50 and 80 micrometers of diameter. So we do then conventional metagenomics. And this is possible because of the work of a lot of people that have been for over 2.5 years developing the technology without knowing whether we would succeed at all. But it is working now, so kudos to everyone. That's been amazing. We are using this laser microdissection microscope, which is in a secret room in the yellow lab. And here in this room, where we have the temperature at about 12 degrees, then we created this case so that the temperature inside can be around 7 degrees, and then the stage has another setup for keeping the slide where the sample stays in about 1, 2, 3 degrees. And this was all custom made for the project by Leica, the manufacturer. And more recently, we have also acquired a Tican dream prep machine for scaling up everything that we are doing, because you need to take into account that each sample in a traditional metagenomic analysis suddenly becomes hundreds of thousands of samples. So we really need to speed this up. And Jonas and Amalia have been working in making this work. Scaling, they are progressing, but automation is never easy unless when you are starting from 10 picograms of DNA. And but we are progressing. That's it. And the very cool thing is that we do observe significant differences between the macro scale analysis and the micro scale. So a lot of the indirect inferences we have done on from the macro scale on microbe interactions, we are quite sure that they are not real. So there are a lot of artifacts because the way we have been analyzing it was what we were able to do with the technology at that time. But when you really zoom in at their scale, microbes are located and probably connected to each other in different ways. So we will get some more information on this, a lot more information in the coming years. The project will be active until the end of 2025, and I'm pretty sure that then there will be data for the next 15 years to analyze this. Related to all these 2 projects, and I'm finishing, we do a lot of development of different procedures, methods, informatic pipelines, R packages, etc. You have an overview of those. HillDeep is for analyzing diversity using Hill numbers, distillr for distilling raw genome annotations into what we call genomic first functional traits that can be easily understood by biologists. And then we are now developing this MetaDeep on using genome scale metabolic network information to quantify microbial interactions and quantify the structure of the microbial communities from that perspective. Then Rafael has been working with Ben Woodruff in Australia to create this tool associated with single M to estimate the fraction of microbial DNA in the samples, which is something that a lot of people thought it would not be possible to do. And then Rafael had this idea. We in the beginning thought it was, this is too easy, this is too naive, this won't work. But then it happens that sometimes the easiest solution is one of the easiest solutions might be the best. So this tool is now available, and it's the papers under review. And then we have 2 main bioinformatic pipeline sets. So then we have the earthology one that is standardized, and then building on what we created on the EHI, then Jorge has been working the last year and a half in creating a suite of multi-omic bioinformatic tools based on SnakeMake and hundreds of different software, and they are all publicly available in the GitHub pages. We also, since half a year ago or so, write everything, all the code of all the analysis that we do in our book down, and we publish it in GitHub pages. So if you go to our GitHub account, you will see a lot of these reports in which all the analysis that we do are rendered into websites where you can see everything and you can redo yourself everything. So I think this is very useful for any people that are willing to work on microbiome stuff, because I know ourselves as well, because 90% of the work is just copy paste in chunks. As we have standardized everything, then everything goes very fast. But 3Domics has its own stuff, EHI its own stuff, and the rest in our group is with the footprint icon. So that was it.